Good day. I am Hannah Magno, together with Rianna Cruz from University of the Philippines, Diliman, and our research is about dominating balanced protein networks in cancer. Our goal is to find the driver proteins in a network to know what mutation of genes or proteins cause diseases, especially in cancer. Given a network, how do we identify the driver proteins? Proteins regulate with other proteins and is referred to as the protein regulatory networks. Proteins interact in various ways, and we denote the positive interaction when a protein upregulates another protein and when it increases a cell sensitivity to the other protein. A negative interaction is when a protein downregulates another protein and decreases its sensitivity. Protein-protein interactions can be represented as a graph with proteins as the nodes and the interactions as the edges. Specifically, we represented the protein regulatory networks as sign graphs. A sign graph is defined as a directed graph where each edge has a positive or negative sign. The sign of the path is the product of the signs of the edges along a path. For example, in a path from vertex 3 to vertex 8, the sign of the edge is -1. From vertex 8 to vertex 9, the sign of the edge is -1, and from vertex 9 to vertex 4, the sign of the edge is -1. And by multiplying all the edges, the sign of the path is -1. Another example in the path from vertex 0 to 9 to 4 to 5 and to 6, the sign of the path is simply 1 by multiplying all the edges. Next, a sign graph is said to be a balanced sign-directed graph if the product of all path signs is positive. In this graph example, all the signs of paths starting from vertex 1 is positive. So this graph is a balanced sign-directed graph. Preliminaries for dominating sets. A dominating set is a subset D of vertex set V of G, where every vertex in V not in D is adjacent to at least one vertex in D. In this graph, the set 1, 3 is a dominating set. It is because every vertex in the graph is adjacent to vertex 1 and 3. Finally, the MCDS or the minimum connected dominating set is a connected dominating set of G with the minimum size. In this graph, the set 1, 2, 3 is the MCDS for it has the minimum number of connected dominating set that is adjacent to every vertex in the graph. The shortest path tree is the tree of shortest paths in graph G and rooted in vertex V. We applied an algorithm to balance the network. To model gene regulatory networks, we use sign-directed graphs because signed edges may model their regulatory interactions. Negative edges model downregulation, while positive edges model other interaction types. We use the shortest path tree to model our gene regulatory networks considering Occam's razor, which tells that if we are given multiple solutions, we must choose the simplest solution. With gene regulatory networks where multiple paths may exist from any protein to another, this leads us to select the shortest path between the two proteins. Our algorithm requires a root node or set of root nodes to model the regulatory function of driver proteins. The relaxed definition of balance in a given network is that it is not completely balanced 100%, and we only balance paths to leaf nodes. The input of the balancing algorithm is a sign-directed graph in a set of root nodes from set V. And the output is a balanced shortest path tree, T of G, rooted at a set of root nodes.
The next algorithm we used in our study was an algorithm to find the minimum connected dominating set of a network using topology-based heuristics. For our methods and data, we use the Senior dataset, which is an open source dataset of protein-protein interactions in the human system, which we divided into test sets, which we implemented our algorithm on, and validation sets, which we compared the results of our algorithm to. From the driver proteins we found, we performed pathway analysis, and from the pathways we got from here, we performed a literature survey to see how many of these pathways actually did have a link with cancer. What we found was that our driver proteins were more topologically significant than the control groups. Here we can see that their betweenness centrality had a higher median range. Their degree centrality also had a higher median range. And they had a comparable consensus with positive controls from other methods. We also found that the driver proteins we found for each cancer enriched pathways with statistical significance that about 80% of the time have been linked with cancer. Here we can see that that's about 10% more pathways than those not enriched by our driver proteins. So from these, we validate that sign graphs can model the regulatory functions of protein networks, and that minimum connected dominating sets and sign graphs can be used to identify some driver proteins of cancers comparable to other computational methods, and that when modeling disease networks, it may be best to incorporate some a priori knowledge. Thank you for listening.